Hi and welcome back. Today we're going to be starting our study of biomolecules and how that goes into food chemistry for area of study 2. And we're going to start with amino acids and proteins. This means that we'll be looking at chapter 15, looking at key food molecules and how they are broken down and synthesized in the body. So biomolecules are essentially organic compounds that occur within living systems. So this is humans and other living systems. They are predominantly made up of hydrogen, oxygen, carbon, and nitrogen. You may have seen this if you've studied biology, where we have CHO and then N. Okay, so when we talked earlier in Unit 3 about fuels and there being nitrogen and other elements present in our coal or our oil, biomolecules are actually where these have come from. Okay, so they are large molecules with specific biological functions. Depending on the proteins or the nature of them, we will see some sulfur, phosphorus, calcium, and other elements that come from particular minerals that are used within our body as well. All biomolecules are formed from condensation reactions. So all the ones that we're going to be looking at are formed from condensation reactions, just like the esters that we've seen previously. So they're going to be the coming together of two molecules to form a larger molecule, giving off 1 unit of water for each condensation. When we break these down during digestion, we see the opposite process of condensation, which is hydrolysis, to produce the small monomers that were previously used to synthesize these. And this is what happens when we get the building blocks for proteins in our body by eating food and things like that. So biomolecules have important roles in our body, including supplying energy to the body, the monomers that we need for growth and repair of organs and tissues. They allow us to move our muscles, the activity of our nervous system and hormonal systems, and elimination of waste. So they are a critical part of chemistry, looking at the chemistry of living systems. There are 5 main types of biomolecules. These are proteins, carbohydrates, fats, oils, which are triglycerides, which we saw with the production of biodiesel, and we will revisit again, otherwise known as lipids, and then vitamins. Nucleic acids and the formation of the DNA biopolymer are also biomolecules. However, the nucleic acids are not part of the study design anymore. So if you're looking at older resources, they will have questions about DNA, but that is not relevant to us. At this point in time, we're going to focus on proteins and where we source them from, how they're made, and then in the next video, we'll look at the structure. So proteins are a type of biopolymer or biomacromolecule, meaning they are very large, that are present in all living cells. Okay, they're fundamental to cell structure and operation, and they pretty much are responsible for carrying out all the processes in our body. They form structural tissues. They can also form functional molecules such as our enzymes and receptors in our body that allow us to regulate body functions. Plants can manufacture all of the monomers for biomolecules, which are all amino acids, all 20 amino acids. Okay, however, animals only synthesize 11 of the 20 amino acids. The 9 amino acids that we are unable to synthesize naturally in our body are referred to as the essential amino acids, and these are what we must obtain from a healthy diet in order to ensure healthy function of our body. There are 2 types of proteins that we see that are categorized according to their structure. These are fibrous proteins, which form structural components of our bodies. Okay, so the dominant secondary structure, which we'll come to talk about later, means that these tend to be insoluble in water. These are really important for structure and support, so we tend to see them in things like keratin, which is our hair and nails, okay, collagen, which help keep our bones joined together with our ligaments and things like that and keep our tissues and skin and tendons flexible and working well. Enzymes tend to be globular, okay? So our enzymes and small proteins used for hormones control and things like that tend to be globular proteins. So fibrous proteins are, as they are described, long and fibrous. They tend to be non-water soluble and responsible for structure. And then we have globular proteins, which form these big, they look like globs. So the names are quite indicative of the way that they looked. All proteins are made up of amino acids. Amino acids form the monomers for the biopolymer that is known as a protein. Okay, so these are monomers that undergo condensation reactions to form the biopolymer. So amino acids are critical, and as we said before, there are 20 amino acids, 9 of which are essential. The other 11 we are able to produce in our body. All amino acids in our body follow the same generic structure. This is, they are 2 amino acids, otherwise known as alpha amino acids. This means that the side chain here is on carbon 2. Remembering we number from the carboxylic acid down, so amino acids which have both a carboxyl group and an amino group. Okay, this means that this central carbon here, where the side chain variation occurs, is on carbon 2. Okay, so every amino acid has a carboxyl and an amino. It is a alpha amino acid containing the carboxyl and the NH2 bonded to the same central atom. So these two are bonded to the same central atom. It is not an alpha amino acid if the carboxyl and the amino are not bonded to carbon 2.
This image shows us the 20 common amino acids, which are also given for you in your data booklet in the last 2 pages. They're generally shown as a combination of skeletal structures and semi-structural. Remembering, if you're asked to draw them, you do need to show all bonds. The amino acids form the monomers of the polymers that are known as proteins. So the properties of the amino acids that go into the protein sequence determine the property of the protein. The dashed lines around here indicate which are the 9 essential amino acids, which are isoleucine, leucine, valine, phenylalanine, tryptophan, histidine, lysine, threonine, and methionine. All the others we're able to synthesize within our body. These 9 we must obtain from our food sources. Aliphatic refers to amino acids that have just CH2-CH3 combinations on their side chain, so are non-polar and have dispersion forces only. Aromatic contain a benzene functional group. This ring here is our benzene. Okay, so all of the aromatic ones have this 6-membered ring with the alternating double bonds. Acidic amino acids are able to donate a proton, so they have a carboxyl on the side chain. In this case, at biological pH, it is typically already protonated, giving us the negative ions. Basic amino acids such as arginine, histidine, and lysine contain amino groups on the end of their side chain. These amino groups are able to act as a base, so they can be protonated in a positive form depending on the pH. Serine and threonine are known as hydroxylic, i.e. they have hydroxyls and are able to form hydrogen bonds. Then we have sulfur-containing amino acids as well, such as methionine and cysteine, which will become important later when we talk about secondary structure. Then the last 2 are asparagine and glutamine. These have amides on their side chains. Okay, they have primary amides on their side chains, giving them the ability to form hydrogen bonds and, again, different functionality. So as we said, we were talking about the R groups. The different properties of amino acids are due to the different R groups, these variable side chains that give each amino acid its unique structure. There are non-polar and polar amino acids. You do need to be able to identify these in a sequence and explain the intermolecular forces that would be occurring between these groups. The non-polar ones are considered to be hydrophobic, that is water hating. So we'll tend to find those on the internal surface of a protein where they are protected from the aqueous-rich environment of the cell. Hydrophilic amino acids are water loving, So they will tend to be on the outer surface of a protein, and we have a high number of these if the protein is water soluble. Here, alanine with just its simple CH3 R group. Okay, this is the R group here. We can see that because we have our amino, our alpha carbon, and our carboxyl, and then the variable side chain here. In this case, we have serine, which is one of our hydroxyl-containing amino acids, with a CH2OH side chain, this variable side chain here. It is important to note that we introduced the idea of chirality during our last organic unit. Chiral groups, remember, chiral atoms have 4 different groups attached to that carbon. All amino acids will be chiral with the exception of glycine. And this is because glycine, its side chain is just, this is our carboxyl, our alpha carbon round to our amino. Okay, so this is our side chain and this is the hydrogen that is usually there. This side chain, because this has 2 hydrogens, it is a non-chiral or achiral amino acid, the only one. All the other amino acids, because of their variable side chain, such as the CH3 on alanine, mean that we have 4 groups attached to the alpha carbon, so they will have enantiomers, they will be chiral. Only one of the enantiomers is used in the body for the production of proteins. So, with our R groups, we also talked about proton donors and proton acceptors. So you need to make note of which amino acids can act as acids and bases within the body. This is important. You will be asked questions about which ones would protonate or drawing the structures at different pHs. Speaking of different pHs, the amino acid structures are unique in that they contain both a carboxyl group, which can act as an acid, and an amino group, which can act as a base. These 2 functional groups mean that the amino acids are amphiprotic, can act as both an acid and a base, but the way they ionize in aqueous solution depends on the pH of the solution. At neutral pH, we can see the proton jumping from the carboxyl down to the amino group. This means that we end up with a negative charge forming on the carboxylate anion. And then the positive charge forming up here on the protonated nitrogen. Okay, so these charges are important. We call this dual-charged form of an amino acid a Zwitter ion. Zwitter ions are present in neutral solutions. They're the main form that will be present in a neutral solution of amino acids. If the solution is acidic, i.e. the pH is going to be less than 7, then this means that we have an excess of H+. Thinking about this being an equilibrium system, this means that this proton is unlikely to be donated. In fact, there is an excess of H+ around, so the system will move to decrease that H+, and we see the protonation of the base, i.e. the amino group will accept a proton becoming positively charged. The opposite occurs with the equilibrium in basic solution. Now we have OH- around, which will react with any H+ to form H2O. This means there isn't extra H+ around to protonate this nitrogen. In fact, the equilibrium will be pushed so that the hydroxyl will donate its proton, and we will see H2O being formed. Okay, so the carboxylic acid will act as an acid and donate its proton. So in basic solutions, we will see the negative ion on the carboxyl of the amino acid. In acidic solutions, we will see the positive ion on the nitrogen as it gains an extra hydrogen. Remembering that pH less than 7 is acidic and that our pH greater than 7 is going to be basic. For protein synthesis, it is a condensation polymerization. Previously, up until this point, we've only talked about addition polymerization where we require a double bond. Proteins are synthesized using condensation polymerization. That is, that the carboxyl of one end of the amino acid will react with the amino of another amino acid. We will produce a molecule of water and the amide functional group. Okay, so this is the amide, and the new bond formed is this one here, is the peptide bond. This is the new bond formed between the carboxyl of one amino acid and the nitrogen of the next amino acid, forming the peptide link. Okay, we eliminate a water molecule in this process, i.e. we produce 1 mole of water for every mole of amino acids that react to form a peptide bond. In order to break this down, we can add water in and facilitate the hydrolysis reaction is the reverse of this. At this point, we're looking at the condensation polymerization for the formation of protein or polypeptide molecules from single amino acid monomers. This is another way of looking at it. Looking at this, it is important to notice that nothing changes with our R groups. During condensation polymerization, our R groups remain the same. It is just the amino from one amino acid and the carboxyl from another amino acid that form the new peptide bond. This means that we can have dipeptides or tripeptides, which are small protein units or peptide units. We can also see polypeptides where we have more than 50 amino acids. Once we get up to this is what we're referring to as proteins. So we can see di, tri, and polypeptides for the sizes of our polymers. Once we reach 50, we're talking about proteins. So let's have a look at some of the questions that might be asked regarding these. Polypeptides are represented as a 3-letter code. This is given in your data booklet. So met refers to methionine, lyse refers to lysine, leucine, arginine, and then methionine. You don't have to memorize these, but it's good if you sort of have an idea of where you would be looking for them. Okay, if we have a look at this and you turn to your data booklet, page 14, I have mine next to me. This question asks us to write the amino acid sequence of the following polypeptide. So we need to identify each of the amino acids. This means we need to figure out which is the side chain, which is the peptide backbone. These are our peptide groups here. Okay, so these are our peptide bonds that are forming. So if we have a look here, we have the alpha amino acid, the alpha carbon of 4 amino acids. So this is a polypeptide. This one here, we have the carboxyl. This would go to an amino here. This is our side chain. That's CH3 side chain. If we look it up, we will find that this is valine. CH2SH, this is going to be cysteine. CHH, this is our smallest amino acid, glycine. And then we have CH2OH, which is serine. So the sequence of the following polypeptide would be valine, cysteine, glycine, and serine, or val, cis, gly, ser. Okay, and then we would just write those together using a capital letter for each of the new 3-letter codes. Again, another type of question. This is a VCAR exam question. We can see that we have a polypeptide. We're told that it's associated with inflammation. And then we're asked to write the abbreviated names, i.e. the 3-letter code for the 2 circled amino acids. If we have a look here, we have our NH2 and our CO making this our alpha carbon. So this will be our side chain. We have a long side chain with the 2 amino up here. This is going to be arginine. Okay, and then we're going to skip over these ones. They're not circled. And then we can see here that it's circling between the NH and C double bond O. This entire amino acid is one of our aromatics, and this is phenylalanine, which means this is Arg, this will be PHE, making A the correct answer for this question. Okay, so the next one is looking at these structures, and you can see that they look a little bit more confusing when we see them all sort of comparing and trying to figure out which one is which. This question has asked us which of the following is a dipeptide made of alpha amino acids? So remember, we have to have the carboxyl and the NH2 bonded to the same carbon, which is carbon 2, our alpha carbon. This means that we're going to look for our carboxyl and amino being bonded to the same carbon. This one is an alpha, but if we have a look here, we have this carboxyl goes to a CH2, CH, and then we have this here. So this is not, because this one here is not our alpha carbon. For B, if we have a look here, here is our peptide bond that has been formed here. This is an alpha carbon, CH, CH2, SH, but this is not, because our amino group is attached here. So it can't be B. With C, we have our peptide amide linkage here, CH-COOH with our side chain. This is an alpha, and then C double bond O going to CH-NH2. This is the alpha, so C appears to be correct. Just double check with D. There's our amide bond, and we can see straight away there's two CH2, so the NH and the COOH are not on the same carbon, so C must be the correct answer. And if we look at the answer, we see, in fact, that is the case. Okay, that's it for protein structure. You should be able to identify them, and you do need to be able to draw them. Our next one will look at how these amino acid sequences fold up and form functional proteins in the body.